We're going to do the notes for Chapter 10, Section 3, Regulating the Cell Cycle. So the cell cycle undergoes normal controls that keep it from dividing too quickly. It keeps it controlling at a normal rate. So these controls turn on and off the cell division. Cyclins are a family of proteins that regulate how often the cell cycle occurs and the timing of that cell cycle. The different combinations of those cyclins signal the activities in the cell cycle, like replicating the DNA, protein synthesis, and then it actually going through the rest of the cell cycle. The proteins that regulate it are internal regulators, external regulators, and growth factors. Internal regulators are your proteins that respond to the events inside the cell, and it allows the cell cycle to proceed once it reaches a certain process. And once certain things have happened in the cell. So on the last slide, what you saw like stoplights. And that's really what these things are. Things don't proceed before they're supposed to. So until they get the green light and certain things happen inside the cell, then they won't proceed through the rest of the cell cycle. External regulators are proteins that respond to events outside the cell. And it would be like maybe you had a cut or something like that and you needed to make more cells. So those would be your external regulators that are saying that, we need to start to speed up or slow down that cell cycle. Growth factors are a type of external regulators that stimulate the growth and cell division. They're very important during embryonic development, and that would be like when babies are being formed and when you have a cut or an injury and it needs to heal. Abnormal cell division is cancer. When you have uncontrolled cell division and uncontrolled cell cycle, that is what cancer is. It's a failure of that cyclin. It's a failure of those systems when your cancer cells begin to grow and are basically growing out of control. Your cancer cells actually spend less time in interphase than normal cells do. We know that we need to suspect cancer when we look at a tissue sample and we see more cells going through mitosis than we normally would. Remember, most of your cells should be in interphase. When we start seeing too many cells dividing, that's when we need to be suspicious that it is cancerous. And cancer cells will kill out and kill your good, healthy tissues and cells by crowding them out. And what you see is it's a mass of cancer cells, also referred to as a tumor. Those tumors block and crowd out the normal cells, and it affects their growth and development. Some causes of cancer are either due to mutations, genetics, but there are various environmental factors that can also affect the occurrence of cancer cells. Substances that are known to cause cancer are called carcinogens. Think about some things that you know are known carcinogens: smoking, radiation. All of those things are known carcinogens. Apoptosis is programmed cell death. You should have certain cells die off. It's a controlled process. We don't want too many being killed off or dying off, and we don't want not enough. Too much or too little apoptosis is what results in many diseases. When we talk about like Alzheimer's, Huntington's, Parkinson's, what you have there is you have too much apoptosis. Too many cells are being killed. Things like cancer and that, too little cells are dying off, and it results in an issue.